All right, folks, the next two sets of slides, the next two videos are just going to be an overview of the four macromolecules that you need to know for biology and a little bit of chemistry thrown in. This should be a review from Gen Bio. Hopefully you talked about the four macromolecules that make up everything. My goal is to make this as short as possible. And let's just get started. So there are four large, what we call biomolecules, basically the four molecules that make up all living things: carbohydrates, lipids, also known as fats, proteins, and nucleic acids. They are considered macromolecules because they're composed of thousands of covalently bonded atoms. Most of them are strings of carbon. That's what you need to know about that. Hopefully you remember that we talk about the simple building blocks of each of these and what they look like in larger forms. You should have talked about it in Gen Bio. Macromolecules are polymers built from monomers. So for each lipid, protein, carbohydrate, you should know what the monomers are, which is the smallest unit, and what the polymer is, which is the large chain unit that makes up the protein or the carbohydrate. Three of the four classes of the organic molecules are polymers: carbohydrates, proteins, and nucleic acids. Your lipids are going to be something different. When we talk about polymers, which are these long chains, they can be built, they can be broken down. When they are being disassembled, they're disassembled by something called hydrolysis, and they are assembled using dehydration. Hopefully, if we look, that this is going to be actually the formation. So dehydration results in the formation of a long polymer. So let's say I'm making a carbohydrate. I am losing water out of that reaction, hence the name dehydration. When I am, hi, come in. Sorry. It's okay. Interrupting people. So dehydration means putting together to make a bigger polymer. Hydrolysis is going to be breaking things down. You are putting water back into the equation, and hydro is going to be the prefix for water. When we talk about how this relates to biology, there's a ton of diversity even between our four categories, which is how we get so much diversity in the grander scheme of things. Those four macromolecules make up everything. First ones we're going to talk about are carbohydrates. These include sugars and the polymers of sugars. The simplest carbohydrates are monosaccharides, or simple sugars, and we put a bunch of monosaccharides together to make polysaccharides. So we would say the monomer is a monosaccharide. It's the smallest unit. The polymers, we call them polysaccharides. So that's a long chain of a carbohydrate, composed by all these sugar building blocks. Monosaccharides, what you need to know from this is that it's usually the molecular formula. So the simplest is 1 carbon to every 2 hydrogens to 1 oxygen. So glucose, for example, is still a 1 to 2 to 1 ratio. It is the most simple monosaccharide, and they can join together to form something much bigger. Also important is these are organic molecules. They are just mostly carbons, hydrogens, and oxygens. So they are considered organic. We classify these by what we call the skeleton, which is the number of carbons, and what we call a carbonyl group, which if you look at on the screen, there is this ring, and then on the top there's this little addition. That is going to determine what function it has. A disaccharide is formed when we have a dehydration, so we lose water, and we join 2 monosaccharides. It creates something we call a glycosidic linkage, but you don't need to know that. What you need to know is we go, monosaccharide is the simplest, disaccharide is going to be 2, putting a bunch of those together will be your polysaccharide. Examples of this: glucose, fructose. Don't really worry about that too much. Again, other examples, maltose and sucrose, are going to be disaccharides because they're made of 2 monosaccharide units. Polysaccharides, these are the polymers, so long chains, and they have storage and structural role. Structural roles. The bonds basically hold energy. Starch, so things in potatoes. This is a storage polysaccharide. It's also in plants. Basically, it is a bunch of glucose molecules linked together in a chain, which is shown in the bottom right of this slide. And so that stores a lot of energy, which is why, for example, like potato is a sustenance food. If you are trying to like just survive, potatoes are a really good source of energy. It's why we saw like the Irish use potatoes as a crop when they were going through like a poor time in their history. Glycogen is another one. This is mostly in the liver and in the muscle cells. So glycogen is the type of energy that you would get from eating meat. Again, it's just a long chain of monosaccharides. Structural polysaccharides are not used to have energy. They are used to provide support. Cellulose is going to be a polysaccharide that we see creating stiffness in plants that allows them to have like a stiff, rigid wall. Cellulose can be eaten. That's why we consume all sorts of plants. It is like what makes a plant have like firm, crunchy texture. That's cellulose. Termites also like to eat it. It's in wood, which is why they like to eat it. So things to think about: polysaccharides in relation to biology. Polysaccharides, so long chains of carbohydrates, show up all over the place. Cellulose in plants and like in things that we ingest. We see chitin, which is another polysaccharide, and it's found in the exoskeletons of arthropods and the structure of fungi. But carbohydrates, a lot of the time we think about as food sources, but when they are structural, they create this like hard surface. And so chitin forms these skeletons. It is also used by surgeons to create thread that they would sew up a wound, and it decomposes after the incision heals. So we've talked about carbohydrates. Lipids are the next type of macromolecule we're going to talk about. Lipids are a diverse group. They are the only class that don't form polymers. The only thing that really unifies them as a group is that they have no affinity for water, right? So they are not soluble in water. They are hydrophobic because they consist of nonpolar covalent bonds, which means they don't have any charge, which means charged molecules like water can't pull them apart. The most important lipids are fats, phospholipids, and steroids. Fats, what we need to know about them is they are a storage molecule. Basically, what you need to know is that the smallest unit, the fatty acid, is going to store energy in its bonds. They do not dissolve in water because they have covalent bonds, which means that they are very strong bonds. They hold a lot of energy, but they don't dissolve into water. When we think about like how this shows up in the real world, we ingest saturated versus unsaturated fats, and it has to do with the bonds. And this is also why some of them are broken down more easily and more readily than others. Presumably, the saturated fats are going to not be as good for you as the unsaturated fats because there's a difference in bonds. We don't really need to know this, but it is useful if you want to use it as an example. Unsaturated fats, again, have one or more double bonds. Makes them more stable. It's what we find in like olive oil and fish fats, and those should technically be better for us. This is not necessary. You can pause here if you want to. Again, trans fats contain more saturated fats, which are not good for us. Just means they don't have double bonds. Again, this is just an example. Don't spend too much time worrying about this. Major functions of fat storage. Basically, we have adipose cells. These are super important because they provide cushion, they provide insulation. We just can sometimes store too much fat, which is when we see obesity showing up, but they do store a lot of energy. Phospholipids are the second type of lipid. These make up the lipid bilayer of cells. That's why they're important. And we've already talked about this, so I'm not going to rehash it. Ask questions if you need them. This is what a single one looks like, but it's not super important. The last type of lipid are your steroids. Steroids are lipids characterized by a carbon skeleton and 4 fused rings. Cholesterol is an example of a steroid. We also know that we have good and bad cholesterol, and hopefully you are already thinking about the fact that probably has to do with bonds, because just like unsaturated and saturated fats, the good and bad cholesterol, as we talk about, is just a structural difference. So those are your first 2 macromolecules. If you switch to the next set of slides, we'll cover the last 2. And make sure you pause, you write down your questions, because we'll do questions and we'll do a quiz on this on Monday.